Mono, di, and polysaccharides, AQA biology. There are a few more key terms than the last video, but once you get your head around it, it's not that much. Like in the last video, we described what a mono, di, and poly was. Now, saccharide just means sugar. So it'd be one monomer of sugar would be monosaccharide, and so on for the rest. Now, carbohydrate is a carbon molecule which is combined with water. So it'd have the structure of carbon atoms, hydrogen atoms, and oxygen atoms. Now, maltose, sucrose, and lactose are all disaccharides. And glucose, fructose, and galactose are all monosaccharides. Glucose plus glucose makes maltose. Sucrose is made up of fructose and glucose, and lactose is made up of galactose and glucose. A reducing sugar is when a substance readily loses an electron to another substance and the other one is reduced. Qualitative is no indication of an exact amount, and that could be, for example, with a reaction with starch. When iodine shows starch, it doesn't show the quantity of starch that is in the molecule, it just shows that the molecule has starch. Semi-qualitative would be, for instance, with Benedict's solution, whereby even though a solution, a positive test would turn brick red, it also has other colours than which it turns. For instance, if there's only a little bit of a reducing sugar, then it turns green. Maybe more or maybe yellow, medium would be brown, until it gets to brick red, which is the where it's got a lot. And then quantitative is a numerical value. For instance, with the rate of reaction, we know. And then a calibration curve is to determine the exact amount of a substance. Monosaccharides. All life is based on carbon. Most substances are made up of carbon-carbon bonds, which are various lengths, and carbon hardly ever bonds with other atoms. Life is based off a small amount of elements. So some examples of monosaccharides are glucose, galactose, and fructose, like I said previously. And a test to test for one of these monosaccharides would be Benedict's. So how Benedict's works is, Benedict's is a blue solution, and when it's added and heated, a positive result with it turning brick red. Now, what is the purpose of monosaccharides? Well, it's a rapid energy source, is readily absorbed, and requires little to no change for use in respiration. Disaccharides. Now, how is a disaccharide formed? Well, like in the previous video where I explained about how two monomers make a di, it's the same with disaccharides. Two monomers of sugar form a disaccharide, and is formed in a condensation reaction, and the bond that is formed between these two monosaccharides is called a glycosidic bond. And in order to break the bond, you need to do a hydrolysis reaction, which if you don't know what that is, you should check out my last video. Now, this is alpha and beta glucose. And the difference between the two is the fact that with alpha glucose, both OH groups are at the bottom. And in beta glucose, they alternate. Now, the carbon in which the OHs are bonded to can either be 1 or 4 due to its structure, because it has 5 carbons and 1 oxygen, in which is in the ring structure. Now, the reason why you can't see these carbons is because of the fact that it's the skeletal formula, and this is just easier to draw in your exam. Polysaccharides. Now, what are some properties of polysaccharides? Well, they're large molecules which can be hydrolyzed to form mono and disaccharides. And an example of monosaccharides are cellulose, starch, glycogen, for example. And cellulose is the main structural component in plants. They make up the cell wall. And they are made from beta glucose. And as these molecules lie flat, the hydrogen bonding that form between the chains creates strong microfibrils. And these are only found in plant cells, as humans can't digest cellulose. Starch. Starch is found in animal cells, is used as an energy source, and contains alpha glucose. It's made up of 20% amylose, which is carbon 1 to 4 bonds, and 80% amylopectin, which is made up of 1 to 6 carbon bonds. Glycogen. Now, this is something that I haven't really seen in exam questions as much, but I don't want you to be caught off guard if it does come up. And glycogen is found in animal cells. And like the reason why I said it doesn't really come up because it comes up a lot in A2. So yeah, anyway, yeah, it is an energy source and it has a branch structure with shorter chains in comparison to starch. Now, how do you test for starch? Well, you add iodine, and from the brown solution, it will turn blue-black if starch is present. Why is starch so useful? Well, starch is insoluble, so you can be stored in the body without it dissolving. And it's compact, so a lot can be stored in a small space. How is glucose concentration measured? Well, you add potassium permanganate, which is a pink solution, and glucose ends up reducing it. And then you'd plot a calibration curve with different concentrations of glucose and measure the time taken for it to turn colorless, showing the rate of reaction. So if you had an unknown solution and you didn't know, you could base it off of the standards and find out where it is on the graph. Why is it important to test for glucose concentration? Well, some people have diabetes, so they need to know the amount of sugar that they're consuming in a meal. Same with wines. Some people like dry wines, other people prefer sweet wines. And if people are going on diets, they don't want to be consuming more sugar than they were before. So if they're advertising low sugars, they actually have to meet that quota. Now, this is all for this lesson. Thank you for listening, and the next lesson will be about lipids.